Hello, everyone, and welcome to the webinar, Marine Genetic Resources, Building Capacities for Ocean Governance. This event is hosted by the Strong High Seas project and is part of a series of events organized by the project dealing with topics around the conservation and sustainable use of marine biodiversity in areas beyond national jurisdiction, including the BBNJ negotiations. This is the second webinar hosted by the Strong High Seas project. The first webinar looked into the topic of marine biodiversity beyond national jurisdiction in the Southeast Pacific and Southeast Atlantic. If you have missed it and are interested in listening to it, you can find it on our website, www.prog-ocean.org/ under the tab Strong High Seas Resources. Before we start, we wanted to give you some information about the Strong High Seas project. This is a five-year project that started in June 2017 and will run until May 2022. It is funded by the German Ministry for the Environment, Nature Conservation and Nuclear Safety through the International Climate Initiative, the ICI. It is coordinated by the IASS, the Institute for Advanced Sustainability Studies, and implemented together with IDRI, the Institute for Sustainable Development and International Relations, BirdLife International, the International Ocean Institute South Africa, UCN, Universidad Católica del Norte, WWF Colombia, and WWF Germany. We work together with the Secretariat of the Permanent Commission for the South Pacific, the CPPS, and the Secretariat of the Abidjan Convention. The objective of the Strong High Seas project is to develop and support new approaches for ocean governance towards improved conservation and sustainable management of marine biodiversity in areas beyond national jurisdiction. The project has three main components. The first one focuses on the Southeast Atlantic, the west coast of Africa, and the Southeast Pacific, the west coast of South America. Here, we try to engage with local stakeholders, governments, the private sector, scientists, and civil society to support integrated management approaches for the conservation and sustainable use of biodiversity and facilitate the delivery of these proposed approaches into relevant regional policy processes. The second component of the project focuses on inter-regional exchange between different marine regions regarding best practices and lessons learned for ocean governance in an effort to support the sharing of information and knowledge. The final component of the project explores links and opportunities for regional governance in an international legally binding instrument on marine biodiversity in the high seas, which is currently being negotiated under the United Nations. Our webinar today will give you the opportunity to learn more about the topic of marine genetic resources, one of the 4 elements negotiated as part of the BBNJ negotiations. As part of this webinar, we will be discussing marine genetic resources from in-situ sampling to digital sequences, as well as the role of marine science and capacity building. We have an hour and a half planned for the webinar today, and we will start with a presentation by Dr. Judith Coburn from the University of the West Indies in Trinidad and Tobago, who will give us an overview on the importance of deep sea biodiversity, exploration tools available to explore the deep seas, and the importance of building capacities and ensuring the transfer of technologies. We will then have a presentation by Professor Marcel Jaspers from the University of Aberdeen in the UK on the long and complex path from the sampling of marine genetic resources to their commercialization. Dr. Harriet Hardin Davis of the University of Wollongong in Australia and Christina Jurdy of IUCN will then comment and discuss the presentations before we open the floor for questions. Please write your questions on the side box during the presentations so we can discuss and answer them during the discussion session. All microphones of participants have been muted, and we will also record the webinar and post it on the Strong High Seas website. At the end of the webinar, you will have the opportunity to fill out a very short survey, and we would be very grateful if you could take a moment to fill it out and give us your feedback. That would help us a lot in the organization of future webinars, and we thank you in advance for your help. And now, without further ado, let's start the webinar with the first presentation entitled Deep Seas Strengthening Marine Science and Building Capacities, which will be delivered by Dr. Judith Coburn. Dr. Coburn is head and senior lecturer at the Department of Life Sciences of University of the West Indies at the St. Augustine campus in Trinidad and Tobago.
Hi, good morning, everyone, from warm and sunny Trinidad and Tobago. It is my pleasure to take you for a dive into the deep ocean. And as we've been introduced, I would like to thank the Strong High Seas, EDOSI, my university, and the organizers of the Strong High Seas project for this opportunity to talk about strengthening marine science and building capacity. The deep oceans are basically greater than what we refer to as greater than 200 meters. Sorry, I seem to be having trouble with getting the slides moving. Darren, let's see, I can't get the slides. I'm sorry, I'm just trying to get the slides to move here. For some reason it's not moving. Sorry. Apologies for that. So this is the exciting deep sea world. It is the last frontier. Resources are running out on land, and the deep sea is the newest sort of area for exploration. It is interesting, it's exciting, and it's still pretty much unknown. Most of the deep ocean, accounts for 45% of our planet, and 61% of the surface is ocean. There are over 300 species have been recognized at hydrothermal vents, and 80% of these, or approximately 80% of these, have been found at no other habitat. It is diversity unimagined, and at the moment considered to be rivaling that of tropical rainforests and coral reefs. The technology that has been developed in the last number of years has been able to give us insights into what we have not been seeing before. So it is fairly new, just under about 50 years or so. It is an expensive technology. It is also challenging to work in these environments, but technology and the development rather of equipment has certainly changed the face of deep sea biology. It has also guided us in terms of what we now know, and in fact what we don't know, so that we now have, we are exposed to new territories, we have to add to our textbook information. We have located coral reefs, cold water corals, which we of course initially thought never existed. And of course the animals are very exciting, very bizarre looking, some of them, extremely colorful, and it really adds to the excitement of the biodiversity that exists in the deep sea. Why is the biodiversity important? Not only do they provide resources, but as I mentioned, we are now becoming accustomed to understanding a lot of how the ecosystems interact, from deep canyons to, we know the resources for manganese nodules and those associated at seamounts, cross, and all the exciting animals that we've discovered at hydrothermal vents, as well as methane seeps. The technology has revealed a new and very, very interesting world for us. But getting back to the functions and services that the deep sea provides, as we all are very aware, we get a lot of resources, fish, shellfish, and traditionally it has always been oil and gas. But at the moment, we are going more into marine genetic resources. There's pharmaceuticals that are now being produced from marine resources and the mineral deposits that are referred to. So those were the rich provisioning aspects of the resources. But there are other functions that we're not, that we won't actually see, but that we know as scientists exist, and those are the supporting functions provided by deep seas: the habitat, the trophic support, the refugia for animals, nursery grounds for the fauna. The exciting pictures that we saw before, we know that there are congregations and special communities in these particular environments. There's a huge regulating function that's provided by the deep sea, and of course, we're very well aware that it allows for the continuous nutrient recycling, bio and geochemical recycling going on in the ocean. And of course, the ocean is a big carbon absorber, carbon dioxide absorber, and therefore in effect sequesters carbon. The biodiversity, as I mentioned, the genetic resources, the biomaterials, and of course, these animals that are living at these deep depths, remember it's extremely cold, it's very deep, it's very high pressures, and the organisms are adapted to these environments. The deep, and a lot of these animals are also blind. So again, that's the adaptation for them occurring in these environments, and they're able to interact and still provide such a huge biodiversity component that we are only now beginning to understand. The deep sea, of course, also provides the opportunity for scientific research. As scientists, and especially as scientists in small island developing states, we need to answer questions. We need to understand the ecosystem, the ecosystem services, and how they are impacted by the different activities that take place in the environment. The deep sea is, of course, affords communication products in that you have the communication cables and so exist there. And of course, we are all in awe when we see the animals, as we did in the earlier slides and on this slide. Hopefully we're all inspired by the deep sea, and we will continue to be. As we speak and as the technology improves, there is already a hunt for resources in international waters. As I referred to, the existence of the minerals, the cobalt, the cross, and manganese nodules. There is conversation going on at the moment, and there will be activities such as deep sea mining and further exploration to retrieve these very valuable resources. Scientific research is core to understanding what is taking place in the deep sea, and in terms of governance, the issues are cross-cutting. Given that the technology is available and is being developed, it still is quite a challenge. But in terms of the different areas that are covered or that we are aware of, so in terms of biodiversity and habitats, we know the Convention on Biological Diversity, and of course the present conversation on areas beyond national jurisdiction. The minerals are covered under United Nations Convention on the Law of the Sea and the International Seabed Authority. Shipping and pollution covered by International Maritime Organization. Fishing by the regional marine management organizations. And as I mentioned, cable, pipelines, and bioprospecting at the moment are unregulated. However, scientists need to understand these cross-cutting issues for governance and for how they relate to each other, and to appreciate how we move forward with it. In this respect, capacity building needs to include mechanisms for active networking, and especially small island developing state scientists. We need to become involved in the larger international coordinated projects, and as they relate to those cross-cutting issues that I mentioned in the earlier slide. Across the globe, having said that, yes, the problems in small island states are quite similar to in developed countries because of the growing nature of science and the improvement in the technology, as I've mentioned. So developing science and the technical expertise is always necessary as a capacity building mechanism. And as I mentioned earlier, the cooperation between the groups so that we appreciate how they relate to each other. The seas are all connected, the ecosystems are connected, and the issues for governance are obviously connected. But we have to think about ways of increasing the funding, and with the larger projects, or we need to seek new ways to find funding, work with industry, work more with our institutions and global institutions to develop projects so that we can all be involved and we can cover the oceans, much of which is still pretty much unexplored. Access through codes of conduct, monitoring, and notification. A lot of this is being discussed at the ABNJ and BBNJ discussion, and a large part of the discussion on the deep sea, the importance and building capacity revolves around how we promote the communication, how we get the messages across, and how we educate everyone on the importance of the deep sea, the resources, and of course, not only those, well, the regulating services, the resources that are provided. And key to all of this, of course, is the impacts of mining and on climate change. The advanced technology and the new exploration tools are key enablers, both for scientists, at the same time for exploration. So the advanced technology does help with providing the information for us and explorers, oil and gas explorers, as well as the mining explorers, the industry, for having access to these resources. So we really need to balance the application of the technology because we want to ensure that we know what's there. We want to ensure that as scientists, we understand the ecosystem functions, and we want to be able to balance what is happening on the development side and the removing and the retrieving of those resources. For example, we need to have training for scientists, and especially scientists coming from less developed countries. And as an example, coming to the Caribbean in 2013 and 2014 was the exploration vessel, the Nautilus. It came to the Caribbean with the latest technological systems, and as seen, the Hercules ROV in the photo. For the first time, we were able, as local Caribbean scientists, be able to participate in such an expedition with these high-resolution seafloor mapping, real-time satellite transmission of data. This really guided us to information that was basically in our backyard in the Caribbean. It's very exciting for local scientists to be trained, and we must think of mechanisms to assist and to continue this kind of development. Capacity building includes technology transfer. There is a lot of technology that's already known, technology with working of the instruments, the computing and the data analysis, and including the biodiversity and taxonomic expertise. We need to know which species we're looking at. And as an example, when the Nautilus, the EV Nautilus came to the Caribbean, it was very interesting because it was for the first time we picked up these extremely large specimens that are on the slide. They were discovered off of Kikamjeni, and they are on record the largest Bathymodiolus mussels that were discovered to date. So being able to be on a cruise, being able to contribute, being able to transfer that technology of being able to retrieve these resources, and sets us up it sets scientists up in a much better capacity, in a much better area, so that we can continue working on the developing and deriving value from marine biodiversity, as it relates, of course, to marine genetic resources, which we will hear more from by the next speaker, Professor Jaspers. But we need to be able to develop the technology and the transfer of technology to be able to set small laboratories up that will work on marine genomics. It is quite expensive. It's quite expensive at the moment for less developed countries, but we need to think about building the partnerships and working on resources and projects that can fund development of this type. As an example, in the Caribbean, and as I mentioned, in terms of partnerships and alliances, this really is a must for us building capacity in areas such as the Caribbean or less developed areas. And coming out of that EV Nautilus project, Dr. Diva, Amon, and myself, we worked with, as you can see, global partnerships, National Geographic, Deep Ocean DOSI, Deep Ocean Stewardship Initiative, Boston University, MIT Media Lab, working in partnerships and alliances towards developing capacity for us. So we were able to initiate this project, My Deep Sea, My Backyard. In other words, exploring our own deep sea backyard using low-cost technology while also building lasting in-country capacity. And I think this is really one of the key objectives of building capacity and training and technology transfer. As scientists, we want to attempt to continue filling those gaps in knowledge. All of this information, all of this gathering of information that we can then use, which will support the enabling the approach, a science-based approach to conservation and sustainable use of biodiversity in not only the deep sea, but in areas beyond national jurisdiction. And with that, I would like to thank you very much.
Thank you very much, Dr. Coburn, for your great presentation on the importance of deep seas ecosystem and the necessity to strengthen scientific research and to build capacities of countries, and particularly developing countries, in better understanding them. For the audience, please do not forget to write your questions on the box on your right-hand side for Dr. Coburn. We will then get to them in the discussion session after the presentations. And now for the second presentation, we have Professor Marcel Jaspers. He's going to present on marine genetic resources from sampling to commercialization. He's Professor of Organic Chemistry and Director of the Marine Biodiscovery Center at the University of Aberdeen in the UK, and has also been involved in the BBNJ process since 2014.
Okay. Trying to get it to do slideshow at the moment. Yes. Okay. Thank you very much from the organizers for putting this seminar together. It's very useful for me to hear as well what Judy had to say. I think it's a very powerful introduction to the importance of marine biodiversity and the importance of technology transfer and capacity building. I wanted to go and start with the concept of marine bioprospecting or marine biodiscovery. That is to take something from the sea, could be a bacterium, an invertebrate, or a fungus, and turning it into a useful product or process. It could be something like a medicine or a research tool, an enzyme, something for personal care or nutrition, or maybe a new biomaterial. Now, in order to be able to use or need to use a marine biodiversity for this, you need to have an advantage over terrestrial resources. So, for instance, superior performance or better economics is important here. So, what is the advantage then? most often we find, I'm trying to make this happen. Okay. Most often we find that besides superior performance and better economics, what we need is to have, and the next slide should come up, but I'm not getting anything at the moment. Yeah, there we go. To have what we observe as unprecedented activity in particular applications. And for enzymes, that might mean a new reactivity or a new biotransformation that can be carried out for an industrial process. For small molecules, it might mean new chemical structures with mechanisms of action against diseases that can't otherwise be treated. And for materials, it might be things like a glue that works reversibly underwater, as has been designed based on mussel byssus threads. As Judy already pointed out, marine biodiversity is absolutely enormous in the oceans, and we know only very little about the marine biodiversity that exists out there. For the divisions of marine life, we expect there are more divisions of life under the water than above the water. And for microbial biodiversity, we have even less of an idea of how much is there. No clear estimate, and we don't know anything about its economic value right now. So again, it's really important to recognize the fact is the state of our ignorance of the marine biodiversity that's out there and how much still remains to be discovered. So the pipeline itself is fairly linear. So we start off with sampling some materials. It could be that we sample a sponge or a sea cucumber or a large animal, or alternatively, we might sample something like a soil sample or a sediment sample from which we grow bacteria. Out of that, we often get either the chemistry directly, we extract the chemicals that are produced by the sponge, for instance, or by the bacterium, or we might go and obtain genetic information on that particular organism, which will tell us what kind of chemistry it might be expected to produce, and then how we can extract that later on. Typically at that stage, the chemicals that have been extracted or produced using bacteria or genetic methods can be tested, and we tend to do things like biological assays against diseases such as cancer, or functional testing, for instance, to find materials that have certain properties that are desirable in industry. And then there's a long road to obtaining a product. It can take many years depending on the market you're going for. So each step may take quite a long time, and there might be periods of waiting. For instance, a sample might be put into a repository. It may take time to get genetic sequence data. It may take time to get the chemistry. It may take time to get the assays done, and it may take time to produce a product. And the thing to remember as well is that for a lot of species identification, it's very difficult. There's very little known about these species. Many of them are new to science, and many of them cannot be identified for lack of taxonomists in that specialized field. What we think about is, again, the kind of planning that Judy talked about, is to plan research cruises. And quite often, they start off with a research plan that goes out to a research funder that owns the ship, for instance, the UK's Natural Environment Research Council. And that plan is then used as part of the application to decide whether or not to fund it. And if the grant is awarded, you may get 2 years notice that you have some time on a ship in 2022, for instance. And as you get closer to the time, there'll be feasibility checks. Can it be done? Is the ship the right type for your research? Can it go to the place you want to go? Are you going to enter any marine protected areas? After the cruise, you'll be able to file a cruise report back to the funder, which contains information on what samples were collected where, under what conditions. It might simply say mud sample from 5,000 meters deep on the South Shetland Trough. So most cruises are still for basic research. There's that freedom of marine scientific research to be carried out, and there's an obligation often to file a cruise report to the funder, but this is not absolute. Some funders do not request this. Some charities don't have that requirement. So how might you accommodate bioprospecting? Well, one of the options is to take your cruise report and have it be updated every year to alert to changes of use. For instance, if a sample was used for taxonomy and then a year later was used for marine biodiscovery, you might want to report that back to the funder. You might expect notification to occur when commercialization is happening. So the opportunity here is really for scientists to obtain global data on cruises that are going out, where they're going, when they're going, what they're collecting, and where the samples are after the end of the cruise. meaning there'll be less overlapping research cruises with the same goals. There'll be more sharing, more cooperation, and more coordination. So collecting materials can be done, as it says over there, if you go to like Trinidad and Tobago, you can collect by hand in shallow seas, but more likely for the open ocean, you'll need something like a large research vessel or an ROV. Sampling devices can be as primitive as a coring device, which simply goes into the sediment and collects a long, thin tube of soil or sediment. It could be a grab like this. It could be a gaseous trawl which collects from the sea floor. It could be a water sampler, as you can see over here, a multi-corer as well. So there's different ways of collecting materials from different backgrounds. Quite often, we have to produce enough material to do the initial testing. And for instance, you see me holding a very large sponge over here from the Arctic, and that will be enough to carry out quite a bit of bioprospecting research because that sponge weighs about 10 kilos. However, more normally, we look at the bacteria that can be grown from either that sponge or from the sediment in the soil and in the sea floor, and grow those bacteria. Like this one here is a bacterium from the Mariana Trench, the deepest part of the oceans, and that same strain is grown here at the bottom in small flasks to produce new chemicals. Quite often, if you want to scale up, you have to go to a facility that allows you to grow it at a much larger scale. The chemistry that we do is typically very linear again, so we do chemical extraction of the product inside the sponge, let's say, or the bacterial extract. We might purify the compounds using techniques like chromatography, and we might identify the compound using sophisticated chemical technology such as nuclear magnetic resonance and mass spectrometry. The next stage might be to do biological testing, and that might be to use, in this case, adherent cancer cells, which we try and kill with our extract or pure compounds to show that it has activity against certain diseases. An alternative is to use genetic sequence data to extract information about the organism using its DNA, and that DNA encodes for the instructions to produce proteins which themselves can produce the compounds we're interested in. So at either stage, that DNA, that sequence information is valuable because it contains information about the proteins, which themselves may be useful for industrial processes, or the production of a compound, which we may be able to do to produce the compound without having to access the original genetic resource again. Now, the problem with all of this is, of course, that each step takes time. So in my own experience, taking it all the way from the origin to a product took about 6 years. So it's not a straightforward process, although the process is accelerating all the time. So the timeline is simple. You sample, and then the discovery and then the clinical stages of the research are such that there's a reducing success rate as you go along, and it can take a very long time, maybe up to 10 years, to discover a drug from these resources. At the beginning, it tends to be universities that take the biggest part in doing this. Then it might be universities and small to medium enterprises that take this on, apply for a patent together, and then go forward. At that point, typically at the stage of phase 1 clinical trials, which are human trials, it will be taken over by a large company who licenses the technology from the small company or from the university. So at the beginning, it is scientific knowledge and data. At the end, it's commercial. At the beginning, we would like to point out this is potential value. Until you actually have a product in the clinic, it doesn't have any actual value. Then it's an important thing to recognize that it takes a long time to do this process. There's a lot of research involved in all the steps on the way there, and it takes some time before it becomes actually commercial. At the moment, there are about 9 products in the clinic. There are 3 in phase 3 clinical trials and about 250 in pre-clinical trials, so animal trials. At the moment, most of those are from shallow reef-dwelling organisms, and most of them are anti-cancer agents, with a few other products as well. And most of these are carried out by small companies and not large pharma at all. The website below is very useful for looking up any current information on this. So the case study here is halovan. This sponge was discovered as Hypochondria okodai in Japan. In 1986, it was published as a chemical structure. In 1996, the chemical was made in a synthetic chemistry lab in Eisai, the company in Japan and New York. Derivatives were synthesized 5 years later, And finally, the product was approved 9 years later by the FDA. So again, you can see the pipeline is 24 years from discovery to product on the market. So again, it's not straightforward. It does make roughly $350 million per year right now, but this is, as I said, one of 9 that's been approved so far, and this is also the most profitable so far. Some examples, for instance, there's a pharmaceutical product at the top that has it comes from a Caribbean sea squid. It's produced by semi-synthesis. The bottom one is a painkiller, produced again by recombinant DNA technology. And then some other products as enzymes that are used for DNA duplication, an enzyme used for the generation of biodiesel, a cosmetic produced from a vent bacterium, and at the bottom there, compounds that can prevent biofilm formation on medical products. So just to say, at any of these stages, we have to consider benefit sharing, and we have to think of the size and the timing of the benefits, the cost and the burden of the benefit sharing to the user, the burden of benefit sharing to the regulator. What is the institutional cost? Who are the beneficiaries? How many are there? The impact of the benefit sharing on the beneficiary, and the timing of the transaction. Some of these points were already made by Judy. So the requirements are then that there must be inclusive. Developing state must be included. We must make sure there's facilitate access for the scientific community so that marine scientific research, which is necessary for marine conservation, is not inhibited. Must be legal certainty, predictability and stability for industry, and enforceability for the regulators. So not so complicated that it's not enforceable. And again, the ideas that we had in our background is that basically at this point at the beginning, once you collect a sample, you just share the metadata and DNA data on the sample, and you share samples that are available. So you get first access to those materials, but if there's enough material for non-competing uses by other scientists, they should be able to have access to it. At this period in time, if you want to develop something into a product, you may need to pay some kind of exclusivity fee to keep it secret from others, and then at the end, there'll be some back-ended monetary benefit sharing that occurs. Our system relies on something called online prior electronic notification, which you'll notice is still mentioned in the current draft treaty. And what that means is it builds on existing good practice. And it uses cruise plans as one of the big starting points, and the cruise reports to be used and shared globally. There will be a minimal data set for each sample. We share materials, but have processes to ensure maximum values are obtained from rare samples. So if there's a sample that only is very small in size, then perhaps it should be used by the originator. But if it's a large sample and can be shared, again, shared for non-competing uses. There should be unique identifiers to allow you to trace materials. So, for instance, if you have a sample identifier and it allows you to identify then also the gene sequences that go with that and any downstream products. What I'm not suggesting is a full track and trace where the materials can be located at any point in time, but rather a system in which the user has the relevant identification materials available. There should be fee-free access to materials and raw data, but for instance, if a sample needs to be sent, it should be at the cost of the user, not of the provider. And this exclusivity period could be sensible for certain types of commercialized research, allowing you to be able to use the materials and the materials before anyone else gets access to them. How this works in science is normally that, for instance, if I have a research that's been done on the crystal structure of an enzyme like this, I deposit it in the database, I get an ID, and then 1 year later, the information is released unless I publish. If I publish, it's released at the same time as the publication. So it's a very clear system, and it's very sensible to do that for data where, again, you may not want other people to compete with you as you're still trying to figure out the last parts of the data for publication. However, the current thinking for digital sequence information is slightly different. Again, what is if you have DNA sequence information on a sample, it should be shared right away. That's normally common practice. So is the right of the data producer the first, or is the right of everyone to have access to that data more important? And currently it seems to be the thinking that data should be shared as soon as it's generated, and that therefore it should be unrestricted use for analysis, interpretation, and publication. Some projects already have eliminated any exclusivity periods. Large data sets can be analyzed by many researchers simultaneously without restriction, without really any serious competition, because the data sets are billions of bits of data. And again, there must be some way to credit the producers with having produced that data in the first place. And this wider sharing tends to be better for science as a whole, because everybody can access the data and use it as long as people have the tools to do that. And quite often those tools are freely available online, and there are many examples of good capacity building online or in country to be able to use that digital sequence information. So finally, just to say, we need to build on good scientific practice, and I'd recommend anybody to look at the paper recently by Muriel Rabon and her colleagues on essentially the FAIR principles. So this is the fact that data must be findable, accessible, interoperable, and reusable. And what that means basically is, if you see the scheme here, from a cruise you get data, metadata, sample data, extraction data, reads, etc. And it all gets shared. And a good database to look at, as an example, is the OBIS database on the right-hand corner, the bottom right-hand corner here. So finally, I'd like to thank all of those who helped me with all the thinking behind this, and some of whom are online just now. But I hope you have plenty of questions. Thank you.
Thank you very much, Professor Jaspers, for your insightful presentation on the complex and very lengthy road from discovering and sampling species to producing a final product. That was very interesting. Thank you. We have received already quite a number of questions from the audience, and thank you very much for that. If anyone else has further questions, please don't hesitate to use the box on the right-hand side of your screen and ask your questions to Dr. Coburn or Professor Jaspers. It will also help us if you just mention who it is directed to. Thank you very much. So now we're starting the second part of the webinar, which is a discussion session. So we will have two short reflections on the presentations by Dr. Coburn and Professor Jaspers. Our first discussant is Dr. Harriet Hardin Davis. She is a NERUS Fellow at the University of Wollongong in Australia, and her work focuses on the role of science and technology in the conservation and sustainable use of marine biodiversity in areas beyond national jurisdiction. So Harriet, the floor is yours.
Thank you very much, Carol. Can you hear me okay?
Yes, we can.
Fantastic. Thank you, Carol, for that introduction, and to the project for inviting me to join this webinar this afternoon. Sincere thanks to Dr. Coburn and Professor Jaspers for that fantastic overview of marine genetic resources. You really brought the process of scientific research to light, and those inspiring pictures and images of deep ocean creatures, which certainly give us a lot of food for thought. I'll just kick off, I think, with two clear reflections that I will presume people will pick up on in their discussions. Firstly, I think one of the key things that really was highlighted from those presentations was the critical role of scientific research in unlocking the potential of marine genetic resources, from the initial access to marine biodiversity through any research and development pipeline. And one of the issues that Professor Jaspers picked up on was the potential role of the scientific research community in facilitating access to genetic resources and indeed enabling benefit sharing. This is something that I think can be discussed further, and I won't dwell on it too much, but just to really reinforce that message that the scientific research community does provide an important basis for the sharing of benefits and for accessing genetic resources, be that through access to data or samples or expertise or technology. And there's a policy brief from the Deep Ocean Stewardship Initiative, which does outline some of this based on the Rabon paper, which really highlights that the scientific community best practices can be further strengthened and streamlined through something like the new BBNJ agreement. And some of the ways that there could be further opportunity to share benefits through the scientific research community would be through things like access to information about cruises. That was something that Professor Jaspers touched on, as well as strengthened and streamlined best practices for data use as well, and access to databases. And these are, I think, opportunities that could be further explored through the BBNJ agreement negotiation process to see how benefit sharing can increase scientific research, but I think really importantly, open up scientific research to the full scientific community all around the world. And that really brings me to the second point that I think was really highlighted in Dr. Cobur's presentation about the huge opportunity to increase the capacity building outcomes from scientific research. The cruise that was mentioned that happened a couple of years ago as being a really successful example, I think is a very interesting food for thought for us to how we can make the most of other cruises that are taking place in regions all around the world, and how perhaps we could do more to involve scientists in those opportunities and really make sure that the capacity building potential is being fulfilled. Ways of sharing information, ways of identifying current capacity, as well as potential capacity building aspirations or needs, and better communicating those needs could be an important first step towards making sure that there's the opportunity for scientists and other stakeholders to be involved in these opportunities right from the very beginning of accessing in through scientific research. And just a final bit of food for thought about benefits and benefit sharing, which we began to touch on at the end of Professor Jaspers's presentation. I think that the presentations we saw today really highlighted that the value of genetic resources is much more than economic value. There is societal value in terms of the potential applications of research and development. There may be cultural value associated with some species. And of course, there's scientific value in advancing knowledge. And so when we think about how that value is translated into benefits, the way we can think about benefit sharing, there's a number of different ways. But of course, the potential monetary benefits is just one piece of the pie. There are also a series of non-monetary benefits in terms of the access to information and knowledge, and those opportunities that were really well highlighted in those presentations. So certainly thinking around the relationship between technology transfer and capacity building and benefit sharing, I think there's further we can go in the BBNJ negotiations to make the most of those opportunities. I'll leave it there and look forward to the discussions, and thank you again for those excellent presentations.
Thank you very much, Dr. Hardin Davis, for your discussion on the critical role of scientific research and the role of the scientific communities in contributing to the access and benefit sharing of marine genetic resources. That was very insightful. I suggest here again to the audience, please write down your questions to Dr. Hardin Davis on the box on the right-hand side of your screen, so we can tackle them in the discussion session just afterwards. And before we start with the Q&A session, I would like to introduce our second discussant for today, Christina Jurdy. She is the Senior High Seas Advisor to the IUCN Global Marine and Polar Program, and also an adjunct professor at the Middlebury Institute of International Studies in Monterey, California. And together with Professor Jaspers, Dr. Coburn, and Dr. Hardin Davis, she is also a member of DOSI, the Deep Ocean Stewardship Initiative. So Christina, the floor is yours. Christina, can you hear us? You might need to unmute your mic.
I'm here. Can you hear me?
Great. Brilliant.
Okay.
Yes, we can hear you. Thank you.
All right. I'll just give my thanks again to ISS and to our previous presenters, Dr. Hardin Davies, Dr. Jaspers, and to Dr. Judy Coburn for their enlightening presentations about the scope and application of marine genetic resources and the issues related to capacity building and technology transfer for scientists, particularly with the deep sea. I would like to take us on a slightly different or complementary tangent. As Carol said, I also teach international marine environmental law out in California, and I've had a chance to get to know some of the scientists who are working on marine genetic resources in a way that is different from the traditional focus that we've had on the exploration of biodiversity for bioprospecting, which is Dr. Jaspers explained, is used to discover natural products that may have medical or other commercial value. What I'm also aware of is that marine genetic resources are the core for a whole new tool called omics, which are being used in many parts of the world to advance understanding and management of the ocean environment. I've got in my hands the NOAA draft omics strategy, and I'll just read a couple little snippets and then show how it can also be used. Which omics, which is OMICS, refers to a suite of advanced methods used to analyze material such as DNA, RNA, proteins, or metabolites. Omics tools have revolutionized biological study, with benefits applied to medicine, agriculture, and other industries. Omics approaches, from DNA sequencing to small molecule analysis, are being harnessed already for a variety of ocean critical needs, at least in the United States. Omics measurements could indicate ecological status, with monitoring used to inform and track the efficacy of management actions by measuring biodiversity, population distribution, food web function, and organism abundance. Innovative techniques such as analysis of environmental DNA, eDNA, characterize life forms that range from microbes to mammals, using a single sample of seawater or sediment without the need for slow sorting and counting methods. Mission applications that demand more detailed biological information include sustaining fisheries, developing aquaculture, combating harmful and invasive organisms, improving seafood forensics and traceability, as well as discovering pharmaceuticals and other beneficial compounds, plus protecting vulnerable species and habitats such as coral that provide essential fish habitat and fuel tourist economies. So in many ways, the omics tools apply to both questions, well, multiple questions of how do we accelerate and enhance access to this DNA, RNA, and other elements, but also how do we advance application and the ability of scientists from around the world in developed and developing countries to advance and apply these new tools and technologies to better monitoring, studying, conserving, and managing the global ocean beyond national jurisdiction. My recommendation is basically that we look through the provisions we currently have in the draft treaty text as they apply to marine genetic resources. There are various options for defining marine genetic resources, which I would suggest would be best to look at in the broadest sense of marine genetic resources of potential or actual value, and then focus on the avenues for building capacity and technologies, working on this together with developed and developing country scientists to really break through the barriers that are blocking our ability to understand functions. This is already being used in the deep sea, a combination of barcoding and metabarcoding and eDNA analysis, water quality analysis, to identify species we've never heard of, to identify how this ecosystem functions, and to identify how it is changing. This really does create an exciting new frontier of science, and I think we could work together to see how we can apply it through the various elements of the draft treaty on access and benefit sharing of marine genetic resources, but also in the capacity building and technology transfer sides to truly revolutionize and transform our abilities to better conserve and sustain our global ocean commons. Thank you. I'll end there.
Thank you very much, Christina, for your great presentation and discussion on the different how to develop, the ability of the scientists worldwide to apply these new tools and how this could be taken into account into a new BBNJ agreement. We have now received many questions on many different topics, and thank you very much to the audience for sharing these with us. So we will now start the Q&A session, and probably start with one question for Dr. Coburn. The question is, has the increasing amount of philanthropy for ocean issues also resulted in more funding for the small island developing states to engage in deep sea research? And also research vessels funded by philanthropists prioritizing researchers from small island developing states? So maybe I can send these two questions to you, Judith, and give you the opportunity to answer. And then what we will do is just have a couple of other questions directed to the other presenters and have their answers to them. So I'm going to unmute you, Judith, and if you want to answer these questions.
Sure. So the question, yes, of course, philanthropy is in fact helping to promote the international cooperation and transfer of technology in the form of training and, of course, the enhanced access to these novel tools. In respect of the actual funding, now, when there is that access and the transfer, let's be clear that there's need for continuation or further development of that the ability training to use the data that may be acquired. So yes, I would say philanthropy has been quite positive in helping to promote the sharing and the cooperation. Having said that, it will always be, because the second part of the question was whether philanthropists are prioritizing researchers from SIDS. I'm not sure that I can actually see or recognize that they are actually just prioritizing researchers, but I like to see it as they're actually engaging researchers from small island developing states and less developed countries. In the old days, there was no need, or there was no connection, there was no collaboration. You could have someone, as I put it, working in your own backyard or in the dead of night, carrying out research, and we're none the wiser. But I'm very pleased to say that that has changed, not only through philanthropy, but other research, global research projects. So it's not just the actual vessel, but projects that have involved less developed islands, less developed scientists rather from less developed states, that we have been able to have access. So yes, it's all positive in that we are now, there is that recognition of the need for sustained capacity building, not only with invitations to come on board, but in further funding and invitations to actually have technology transfer continued in laboratory sessions and in laboratories abroad, and to some extent developing one's capacity of developing a lab in one's own territory as well. Thank you.
Thank you very much, Judith, for your answer. We've received two questions for Marcel Jaspers. So it's about the sampling of the future, sampling, of marine genetic resources. And the question is whether or not they need to be sampled in situ, and how to keep them alive. What happens if these samples come from areas under, big pressure? And then the second one is what is the likelihood of actually finding something? So knowing that the deep seas or the areas beyond national jurisdiction are so, wide and big, how can you do it as a scientist or, a company? How can you actually say that you're going out to try to find a specific sample? How can you be sure that you will find something that will be of value to science? So I'm going to give you the mic. Okay. Marcel, yeah, if you want to answer.
So the first part of the question was really about keeping the sample alive. And that is technically very challenging if you have to do that. So you could, for instance, get a sea cucumber from the very deep and try and keep it under pressure up to the surface. There are only 1 or 2 facilities globally that can achieve that, and JAMSTEC in Japan is one of them. And that's very specialized also to transfer from the pressurized vessel back to somewhere you can analyze the material. So quite often, when you collect a specimen, it is taken up to the surface without changing whilst letting it change pressure. As it goes up to the surface, it goes to lower pressure, and that would then essentially destroy the organism or somehow degrade the quality that you might have of the tissues. However, the DNA would still be intact sufficiently for you to be able to work on that. With sediment samples, we tend not to worry too much, although there are now some sampling devices that can sample down to at least 3,000 meters under pressure and bring it back up to the surface. But again, that's very challenging to do the transference and then to cultivate bacteria under high pressure. So again, it's a real challenge to do all of that. So we tend to rely on the fact that bacteria are pretty hardy, and that they may survive. On the way to the surface, or there may be spores of the organisms or some kind of dormant form that we can reactivate when it comes to the surface. So again, all of these things are pragmatic based on equipment available. If you have a deep sea corer and you can't change the pressure, then you just bring it to the surface and hope for the best. In terms of actually hitting something, I think the question can be interpreted in 2 ways. Would I hit something I didn't want, or hit something I did want? If I'm fortunate enough to have an ROV, I would be able to take it to a location where I might suspect there are high biodiversity, such as, for instance, a hydrothermal vent field. Then I could go and look there with my ROV and make sure I could collect exactly what I wanted. However, if I'm using a coring device, and dropping it straight down 9,000 meters to the sea floor, I have very little guidance. It may well be that it drifts a kilometer east or west or north or south as it's being dropped. So again, it's a wide-ish area in which you might be collecting materials, and then you hope that it actually collects something at all. It drives into some soft sediment rather than hits a hard rock. So again, these are very challenging things to do and collect. So really, marine genetic resources from the deep sea are incredibly valuable because they're so hard to get a hold of and so expensive to get a hold of.
Thank you very much, Professor Jaspers, for your answer. I have another one for you, which also goes along the same lines, I believe. So the person is asking if or what type of marine genetic resources have been successfully commercialized to date, if these organisms are already known to science, or if you've already also discovered new recently discovered organisms that can be then considered for future commercialization, and how far away from the coasts are most of these species found? I think the question also refers to whether or not these are sampled within national jurisdiction, or if they are also sampled in areas beyond national jurisdiction. So I'm going to leave it at that for the moment. I'm going to give you the mic. Yeah.
Okay. You go. Thank you very much. That's a good question. The ones that have been commercialized to date, most of them, the species were identified after the organism had been extracted, the chemistry identified, and even sometimes it takes a year or two after it's got to the stage of animal trials and things like that, because marine invertebrate taxonomy is very hard to do. There's very few people globally who are able to do specialized identification of something like ascidians or sponges or soft corals, etc. So that's why it can often take time. So quite often it might be identified to family level on site. You might get a genus identification fairly quickly, but the species may be a new description of the species. So again, if it's poorly known organisms, they're often from well-known types, mainly at the moment for whole organisms, sponges, ascidians, soft corals are the main sources of marine natural products that we look for drugs from. In terms of bacteria, they tend to be the rarer forms of actinomycetes, so bacteria of a certain clade that are known on soil, on land, to produce lots of interesting chemistry. But people are now beginning to look in unique locations. For instance, Jan Peel's work on the tectomicrobia has shown that there's a whole clade of bacteria that grows inside sponges that are responsible for the production of much of the chemistry that we observe from them. So again, the science, the chemistry almost led the discovery of new chemistry, led into the discovery of new biology, and it's quite common for it to happen that way around. In terms of how remote the source species are, most of them are coastal reef organisms, but there are some that are deeper. For instance, haloven was obtained from a deep sea Japanese sponge. But remote in terms of taxonomy means, are they different species or phyla? And most of them are different phyla, so they're as far apart as we can think of. So there might be different animal species such as an ascidian, a sea squirt, or a sponge, or a mollusk. Those are all things from which these drugs have been derived. So in terms of the answer to the last part of the question, are we going towards more remote organisms or recently identified species? People are trying to go for more exotic, extreme locations. Might be the deep sea, might be hot vents, to try and identify organisms that are unique in terms of what they can produce. And quite often that's identified using genetic information before they even start doing the chemistry. So you might say this is a unique species. It contains the possibility of producing 10 new metabolites and 10 new enzymes that we are interested in. So that's what's happening right now. But people are still looking in well-trodden locations and finding new things. And again, that's also very exciting. So inside a sponge that had been studied for years and years and years, they're finding a new phylum altogether. And again, that is something we have to recognize, that we're going to keep discovering new things, even in hotly contested and hotly investigated locations.
Thank you very much, Professor Jaspers, for your great answer. There's one question for Dr. Hardin Davis. Given everything that you've told us about the importance of scientific research and the important role of the scientific community in access and benefit sharing of marine genetic resources, the question is around the role of the upcoming United Nations Decade of Science, which will start in 2021 and go until 2030. What do you think could be the role of the decade in supporting implementing or helping scientists across the world to have a stronger voice or provide more information on marine genetic resources and supporting the implementation of the BBNJ agreement? We're going to give you the mic.
Thank you so much, Carol, for that question. I certainly think, in my opinion, that the UN Decade of Ocean Science for Sustainable Development is a critical opportunity to mobilize some of the scientific research and also some of the supporting institutional architecture that could be necessary in order to upscale some of the capacity building initiatives that we heard about. The Decades really galvanizes activities. It provides a call to action and could be a platform that links up different collaborators and helps to focus and concentrate efforts. For example, one of the thematic areas of the Decade is a healthy and resilient ocean. And certainly increasing scientific knowledge of biodiversity beyond national jurisdiction has been one of the key sort of flagship initiatives that was mentioned in the very early discussions around the decade. So that's not to say that there is a project in the works yet, but it's certainly an area which has been discussed as being a priority. And I think that we can certainly hope and contribute to discussions that are trying to increase scientific activity and mobilize new programs that will advance scientific knowledge of biodiversity beyond national jurisdiction. Another important area for the decade is a thematic focus on a transparent and accessible ocean. And a key part of that are things like how can we create much better access to data for those that need it? How can we improve the coordination between capacity building initiatives that currently exist? And how can we upscale those types of projects so that they are more accessible and meeting the needs that are identified by the various scientists, states and other stakeholders. Some of the things that are going on right now which provide opportunities to shape those discussions under the decade are the regional planning meetings. The global planning meeting will be held in March. And of course, there'll also be synergistic discussions going into the June UN Ocean Conference. So I think the decade's a really important opportunity to develop new programs and portfolios of work that could help to really get initiatives off the ground and could link really nicely with some of the legal and policy developments happening under the BBNJ agreement. I think there's a lot of supportive opportunity there.
Thank you very much, Dr. Hardin Davis, for your answer. The next question is for Christina Juddy, and it's about the current version of the BBNJ treaty, the one that was published recently by the president of the conference. And the question is about whether or not this version of the draft treaty still imposes the regulatory burden for documenting and tracking the use of marine genetic resource samples on the marine scientific research community, rather than on those who might eventually profit from the commercial exploitation of marine genetic resources. So Christina, we're gonna give you the mic.
Hi, that's a tough question, and I think I would defer to the scientists on that. Just quickly, I would say that because the benefits from this type of biodiscovery operation are so unclear, especially as they're going to be applied by commercial developments, then I think it is difficult to you don't want to impose burdens on the scientific research and application, and you know, the requirement to track and trace and do the other things you want to with it. If you're going to be extracting commercial benefits, monetary benefits, then yes, you would want to wait for the end user. But with the uncertainty, I think it's really hard to tag it on down. That's rather vague, but I can't see the whole question. So I'll leave it over to Marcel and others to answer more fully.
Can you hear me? I'll answer that question a little bit as well. So it's a very good question, because what it gets to is whether or not we desire a complete track and trace system for marine genetic resources, or whether traceability is enough. And the distinction is, in my mind, track and trace requires you to know where the material is at all times, so that you can find it. So like a parcel being delivered by Amazon or something. On the other side, traceability means that I can find out where the sample came from, given some parameters about the materials. It might be, for instance, a unique identifier that's given to the sample at the beginning, that the end user has to then has the obligation to show that they have that information in order to be able to commercialize it. At the beginning, scientists really need to have a lot of the information that arises from marine genetic resource collection anyway. I mean, that's good scientific practice. So GPS coordinates, what type of sample, where it was collected, when, what day, what the temperature was, the standard metadata. Then it comes to the surface, that data should be deposited in a searchable location. So the FAIR principles should apply to that data. so that somebody else knows that you've got that material and could potentially access it if they wanted to. And again, scientifically, it makes sense to link that information, your unique identifier and your location, to, for instance, genetic sequence information or digital sequence information, whatever you want to call it. But beyond the need for scientific traceability like that, there is, in my mind, better not to have a full track and trace as it goes through commercial enterprises, but rather have those enterprises take the burden and make sure that the traceability is still possible, but they have to take the onus rather than the scientists. That would be my position on that.
Thank you very much. I have another question for Dr. Jaspers. And the question is, what happens with the patents of the products that are the result of the genetic research, the ones that you gather from deep seas organisms? So I'm going to give you the mic back. Oh, yeah.
Yep. Okay.
Thank you.
So there's a question. There's two questions that are very much related, and it's that one plus another one that talks about the embargo and intellectual property and who owns the property rights on MGRs. I'm not an intellectual property lawyer. I do work quite closely with one here in Aberdeen called Abby Brown, and she's been trying to tell me what should happen and how we should do this. The answer to that question is simply that I don't believe that the actual genetic sequence information or digital sequence information should be patentable in it by itself. There needs to be some clever transformation of that DNA sequence information or the materials that you have in order to make it patentable, so that then at that point there's a transition from the information itself to a product or a process that can be developed further on which you apply for the patent. What I would like to see at the end is to make sure that at the end of the product development, again, it's traceable back to its origin, so hence the unique identifier, and that there is a royalty paid back into a central fund for ocean conservation, for instance. So that would be my feeling, and that perhaps you could have that royalty paid into a central fund to be a percentage based on the sector. For instance, pharmaceuticals might be 3% and cosmetics might be 10%. Those are just ideas that I'm throwing out there, but those are the kind of ways you might be able to work this so that it doesn't disincentivize the innovators and gives them a chance to do something with the materials and then also give back to that central fund for ocean conservation.
Thank you very much, Marcel. I have one more question for Christina, and I thought also Harriet, if you want to say something afterwards. The question is about what are the key questions around marine genetic resources that are currently under debate at the BBNJ negotiations and for this new draft that has just recently come out, and how are these progressing? So maybe if I can give you the words first, Christina, and then if Harriet, if you want to add something to it afterwards, I'll give you the mic as well.
I'm going to defer to Harriet and Marcel and Judy on this one. As I said, I'm following this, I guess I was muted at the time. I'm following this mainly from the focus on conservation benefits of marine genetic resources, and would be looking at what the obstacles are to its application, and that's the potential role of capacity building and technology transfer in accelerating its use. So Harriet, over to you for the tough questions.
Hi, thank you, Carol and Christina. That's a really good question.
But thank you, Christina. If you want to say something to it, Harriet?
Yes, can you hear me okay?
Yes.
Great, thank you. I was muted, sorry. Thank you for those questions. It's a really important point. I'd say that as we look at the revised copy of the draft BBNJ text that was released at the end of November, many of the provisions relating to marine genetic resources do still appear in brackets and often have several brackets within those brackets. So I think the short answer is a lot is still on the table, but let me highlight a couple of things. There's still, I think, some discussion regarding the definition of both marine genetic resources and the terms relating to the use of those resources. One of the issues that this speaks to is the scope of our application. There was some discussion, for example, at the last negotiation as to whether this would apply to samples of a resource that have been collected, whether it was samples of a resource that was still in situ, and whether it would also apply to the digital information, be that for example, genetic sequence data that was derived from those samples. So there's certainly still a lot of discussions around the scope. There's also still some discussions about how to make a distinction between the collection of a resource which is for scientific research purposes, including potential biodiscovery or others, or for something which is more of a extractive industry such as fishing. More sort of substantively, there's still discussions ongoing relating to the sharing of benefits and at what point that could or should begin. But I would highlight here that whilst there are many questions that are still under discussion with regards to the draft text, certainly if we look at the benefit sharing draft text that relates to genetic resources in terms of scientific research, capacity building, transfer of technology, access to data, there's a very strong correlation there with the items listed in the annex on technology transfer and the part on technology transfer. So I think certainly there is a foundation there and the building blocks for consensus on benefit sharing that will empower countries with the data and knowledge and skills and tools to understand and conserve and sustainably use biodiversity, albeit with some items for discussion that remain to get there. I hope that helps.
Thank you very much, Harriet. That was really helpful. We have now 3 more questions until the end of the webinar. So I have 2 for Dr. Coburn and then 1 for Professor Jaspers. So the 2 ones to Dr. Coburn are, the first one is, how can we take advantage of the growth in scientific knowledge that hydrothermal vents offer, while ensuring that this important scientific research does not destroy the very ecosystem from which that knowledge may potentially come from? And then the second question is there an example of marine genetic resources associated with minerals? So I'm going to give you the mic. Yeah.
Sure. Hi. Sure. Good question on the scientific research. Most deep sea scientific research is actually non-extractive and therefore by extension non-destructive, and it's one of the sort of core focus that we tend to have in deep sea and marine biology for that matter. So while we're exploring and discovering, we tend not to be actually removing and extracting. Having said that, as in all other environments, if you're using machinery and equipment and tools, the high-tech tools that we are using, the ROVs, and there may be some minor disturbance to the environment, but certainly it's minimal compared to those activities like deep sea mining and extractive, any sort of extractive activity that's going on, the oil and gas exploration and so on in these areas. So suffice it to say that good science endeavors to ensure that we're not actually destroying what we're surveying. The last question that was asked in terms of if there are marine genetic resources associated to minerals, at the moment, I can say that, as Marcel indicated, if there are sediments and microbes that are obviously associated with the mineral that's being extracted, there will be a potential of a marine genetic resource that can be actually extracted. But at the moment, I am not actually aware of any discovery that was made directly associated with minerals. But perhaps Marcel has an idea on that as well. Thank you.
Thank you very much, Judith. Marcel, do you want to add something about the association of MGRs and minerals, maybe from your perspective?
So it's a tricky one. Certainly, if we're looking at how those minerals are treated, for instance, it is possible that the manganese nodules, for instance, have been grown, in fact, by bacterial intervention rather than just by inorganic chemistry. On the other side, deep seabed mining could generate a huge dust plume, which may upset the upper ocean biodiversity assemblage. So again, I'm not really sure what the question was driving at, but those are two possible interpretations.
Thank you very much for that. And I have one more question for you. It's about one of the slides that you had showing the commercialization pipeline. And the question is, would the figure look the same if other commercial users would be looked at, for instance, enzymes? And would the success rate still be so slow? And would or are big companies engaged from the beginning, or do they come in, later throughout the pipeline? Yeah, I'll leave it at that.
Yes, so I saw that question. It's a very good question. So the good news on the one side is that the pharmaceutical industry is incredibly good at putting their data online, and we know for regulatory reasons what the success rate is from the beginning to end, and that's important. However, for other industries, it's much more difficult to obtain the information on how much commercialization there is of, for instance, enzymes from areas beyond national jurisdiction for commercial purposes. We do know that it's a very big playing field. Potentially, industrial biotechnology is a $1 trillion industry. But if you think about that, break it down into the enzymes that might be useful. For instance, let's say cold-adapted enzymes are important. You would know where to search. The Arctic or the Antarctic would allow you to find bacteria that grow in cold water. Then you would take their genomes and identify potential types of genes that would be of interest to you for instance, an enzyme for washing powder or laundry detergent. That would then allow you to find potential enzymes, which you can still have to modify to make them into a product. But again, the success rates are likely to be higher than for drug discovery. But you have also so much choice because you don't necessarily just have to look at the collections. You could look at the databases online to obtain that information. And I recommend that if you have a chance to look at the recent study on databases in digital sequence information. There's a case study there about Novozymes, which is a Danish large conglomerate of large company looking at enzymes for various purposes, including laundry detergent and industrial applications. And they've stated publicly now that they 50% of their data is open access data from the databases. The other 50% is data they keep secret to themselves from materials they've collected around the globe. Again, so large companies do get involved and do gain a large amount of profit probably from this, but we don't know how big it is because it's difficult to obtain that information. And on the other side, there are SMEs still working on enzymes, but highly specialized ones. And an example is Arctic Zymes up in Tromsø in Norway, and they have a very successful pipeline of products for DNA manipulation, for instance, in anthropology and archaeology. And again, so there's lots and lots of applications that are possible, but what needs to be done, I think, is a research project that goes and actually finds out how many industries are involved in this discovery and how many are making substantial profits out of that one. And again, that's not going to be a trivial exercise, but I think it's important to get that information together before we make a decision.
Thank you so much, Marcel, for tackling this difficult question. So this brings us to the end of today's webinar. I'd like to thank you all very much for your great questions and this great discussion. We've received many questions, and we didn't manage to tackle everything during this webinar. So apologies if we have not answered your particular question, but hopefully the discussion was helpful and also enlightful. I would like to thank all the presenters for their great presentations and also for taking the time to share their knowledge with us, and also to all participants for participating in this webinar. And we hope that it was interesting and useful to your work. So if you want to know more about the Strong High Seas project, or if you have any questions, please feel free to contact us at the email address that you can see on the background slide, which is Strong High Seas in one word at iass-potsdam.de. The recording of the webinar and the PowerPoint slides will be shared with all participants. And please also don't forget to fill out the short survey after the webinar ends. So thank you again all very much for your participation, and we wish you all happy holidays and a nice rest of your day. Thank you. Bye.